Thank you very much. It's a great pleasure to be here again this year. It's been a few years since I attended this meeting, so it's been great to hear the progress that's being made in this field, which is considerable. I'm going to spend the next 20 minutes or so talking through some of the research that we've been doing in Newcastle in the northeast of England. But I wanted just to start off by taking a step from the last speaker's talk, really just to underline to you the considerable overlap that there is across a range of different chronic diseases. Chronic disease, according to the World Health Organization, is going to be one of the greatest challenges that faces healthcare systems. And up to 50% of the majority of patients with chronic disease experience the symptom of fatigue. And in Newcastle, we have a consortium that's recently been given status as a university research centre, where we look at fatigue across a range of different chronic diseases, including CFSME. And that allows us to take methodologies and knowledge that we learn from one chronic disease and the symptoms that affect those patients and apply them very quickly to increasing knowledge in other clinical areas. So, for example, diseases that you may not automatically associate with symptoms such as fatigue, like chronic obstructive pulmonary disease, heart failure, type 2 diabetes, are areas where we're beginning to increase our understanding of the symptoms experienced by the patients that we see. And that stems largely from this piece of work that we published in QJM a few years ago, where we used the fatigue impact score and applied that to a range of different cohorts of patients with CFSME and fatigue-associated chronic diseases, and then looked at the experience lived by those individuals and how they perceived the symptoms that they were experiencing. And the long and the short of it is that these patients' fatigue is the same whether you have CFSME or fatigue-associated chronic disease. And that's important when we're advancing knowledge in a range of different diseases. For example, at the moment we have a rituximab trial for fatigue in PBC in Newcastle. So any knowledge that we learn from that particular trial has direct applicability to other fatigue-associated chronic diseases, including CFSME. For those of you that don't know me, I'm interested in the autonomic nervous system, and this is here just illustrating the cardiovascular system, which is innervated by the autonomic nervous system and has intimate control over how blood is distributed around our bodies. At the centre we have the heart, which is the major pump that pumps the blood around the body. At the bottom are our legs. Here at the top is our brain. And physiologically we're challenged because of where our heart is. And that's happened evolutionary over time, where we've moved from being Neanderthal man with our heart and our head in line with each other to where we now have the upright posture with our heart at the centre, pumping consistently blood to our brain against gravity. Our autonomic nervous system, when we stand up, detects that drop in blood pressure that happens as blood drops into our legs. So as I'm standing here, 700 mils of blood has dropped into my legs, and as a consequence of that, there's a microsecond response where the baroreceptors in the arch of my aorta will detect that drop in blood pressure and send via my autonomic nervous system, the parasympathetic branch, an impulse to my brain, which in my brainstem then activates my sympathetic nervous system and tells my cardiovascular system to make my heart go a little bit faster and to make my peripheral blood vessels constrict in order to keep the blood pumping to my brain, which is conserved at all, by all measures. And what that generates is a head of steam, the blood pressure. And the blood pressure is what keeps the blood flowing round our body. And if at the extreme end of things that head of steam isn't high enough to keep my brain perfused with blood, then I'll black out or have syncope. If in the more middle range of things there's not enough blood going to my brain, I'll feel dizzy and lightheaded when I stand up. And at the very subtle end of things, if that head of steam isn't high enough to get the blood to my brain, my heart, or my muscles, that's what I believe manifests as the symptom of fatigue. And if there's a slight dysynchrony between those microsecond responses of my autonomic nervous system in response to manoeuvres like standing up or a Valsalva manoeuvre where I blow against a hard force, then if those responses don't happen to keep my brain, my heart, my muscles perfused with blood, then that's what manifests as dysautonomia. So if my hypothesis is right, and part of the experience of CFSME is related to problems with the autonomic nervous system and dysautonomia, then we would expect that people would have symptoms of dizziness and lightheadedness or blacking out. And we can quantify that very easily in the clinic using a measure called the orthostatic grading scale. And when we've applied that measure across a range of different chronic disease cohorts, you can see there's a wide spread of results, with 4 being indicative of orthostatic intolerance. So you can see in fatigue-associated diseases like fatty liver disease, the autoimmune disease primary biliary cirrhosis, there's a similar spread of values. And when we look cumulatively at that, the long and the short of it is that in patients with CFSME, almost 90% of patients will suffer from orthostatic intolerance, so symptoms of autonomic dysfunction when they stand up. And the fatigue-associated diseases of fatty liver disease and primary biliary cirrhosis will also have a significant proportion of patients with autonomic symptoms. And importantly, in every disease that we've studied to date, fatigue severity associates with the severity of autonomic symptoms, with more symptoms of fatigue being associated with a greater autonomic symptom burden. And that led us to suggest that in patients with CFSME, there was a form of fatigue within that diagnostic group called dysautonomia-associated fatiguing syndrome, and that if you took a cohort of patients with chronic disease, a proportion of whom would be fatigued and a proportion non-fatigued, that in the fatigued group, a subgroup would have a similar dysautonomia-associated fatigue. Going back to something that we discussed at length yesterday at the researchers' meeting about the stratification of patients who were put together into this diagnostic group of CFSME, and how important it is to begin to tease out the different diseases that might be enveloped together in this one big block. So that's symptoms. What about objective measurements of the autonomic nervous system? Well, the simplest one is to just measure that head of steam, the blood pressure. And we can do that using ambulatory devices. So this is a 24-hour recording of blood pressure, and expressed here as mean systolic blood pressure over 24 hours. This is the fatigue-associated disease PBC, and over 100 patients with CFSME and matched sedentary controls of the same age and sex. And you can see that the head of steam in patients with CFSME is significantly lower over 24 hours compared to their matched controls. And this is just one slide that illustrates a decade of my life, but essentially what it's here to illustrate is that if we use objective measures of autonomic function, in this case something called heart rate variability, using whichever modality of autonomic function we use in our experiments, that we consistently find that there are autonomic abnormalities in those who are fatigued and have CFSME compared to non-fatigued chronic disease groups or to controls. So now this looks like an instrument of torture, but actually what it is something called a tilt table. And it's something that we use as an evidence-based assessment tool in our syncope clinic. So people who come to us who drop their blood pressure and present with syncopal attacks, this is an investigation that we perform routinely in this patient group. It looks like a bed, but it's actually a table, and it brings people to standing on the platform here in a standardized way. And we're able to look using continuous heart rate and blood pressure. In our unit, we use a system called the task force. and we're able to look for subtle changes in heart rate and blood pressure in response to the stress of standing. And when we've applied that tool to patients with CFS-ME, and this is a cohort of 64 patients who've been tortured by my tilt table, and 64 matched controls, what this table illustrates for us is that significantly more people with a diagnosis of CFS-ME will have neurally mediated hypotension, i.e. a tendency to drop their blood pressure when they are tilted. And at the bottom here is a group who have a condition called positional tachycardia syndrome, which is a recognized form of dysautonomia where individuals, when they abort to a standing position, will have a tachycardia to above 120 beats per minute or an increase of 30 beats per minute on assuming that upright position. And what we are consistently finding is that up to a third of patients who have a diagnosis of CFS-ME, if they are formally tested in an autonomic protocol, that they will have this positional tachycardia syndrome. And the important message with that is that there are treatments that are available and evidence-based for positional tachycardia syndrome. So what might the mechanisms be for these abnormalities in autonomic function that we're finding? Well, it may be that they're related to brain abnormalities, and we've heard an awful lot about abnormalities of the brain that we find in patients with fatigue-associated diseases. But there may equally be downstream abnormalities of that vascular system. Perhaps people are putting volumes of blood in places that they shouldn't be, and as a result, the head of steam is not as high as it ought to be. And I'm just going to give you some initial results from individual studies that we've done and largely published. But at the moment, we're just about to start the analysis of our comprehensive assessment funded by the MRC, where we've been doing all of these experiments in the same individuals. But to give you a feel of some of the things that we're looking at, we've been doing brain MRI, and I show this one because this is my brain, which is why I put my hand up earlier on when we were asked the question. And brain MRI, looking at cerebral blood flow measurements while individuals perform the stressor of the autonomic nervous system called a Valsalva maneuver. So we get people to blow against a closed glottis for 15 seconds, which we know drops their systemic blood pressure and has a very well-recognized and well-characterized change in systolic blood pressure in the periphery. And what we're looking at is what happens to, at the same time, in terms of their cerebral blood flow. And we've performed a battery of neuropsychometric testing in patients, and this is just one of those measures to illustrate how we're finding abnormalities in cognitive performance on cognitive testing. And when we look at the shifts in blood flow through the Valsalva between the different phases, what we're able to show in this initial pilot study is that there are relationships between performance on cognitive testing and these changes in cerebral blood flow in response to the autonomic stressor of a Valsalva. So all in all, that suggests that there may be some part that the brain plays in these autonomic abnormalities that we're finding in CFS-ME patients. We've also looked downstream with a particular interest in muscle function in patients with fatigue-associated diseases and CFS-ME. And we've been performing MR spectroscopy in patients while they've been exercising in our MR scanner. And we're able to detect changes in pH during bouts of exercise and look at how acid recovers between bouts of exercise. And when we do this, what we've been able to see is that there are huge amounts of acid accumulate in the muscles of CFS-ME patients when they exercise, and between bouts of exercise, they really struggle to get rid of acid between bouts. And the important thing for somebody like me who's interested in autonomic dysfunction is that the severity of these abnormalities associates with the degree of autonomic dysfunction. And we speculate that this is partly related to outflow of runoff of blood from the muscles in response to exercise and its regulation by the autonomic nervous system. We've taken muscle biopsies from those people who have participated in the MRS studies, and we can now grow those muscle cells in the laboratory. And using a system called the CPACs, we're able to exercise those muscle cells that we've grown, so allowing us to replicate what we've seen in the MRS studies in the laboratory. And it looks very, very subtle to you, I suspect, but you've no idea how excited Audrey was when she was able to visualize these muscles contracting here, confirming that we were actually exercising the muscle cells that we were growing in the laboratory. And we've developed nanosensors that we know will reliably go across the cell walls of the muscle cells and fluoresce at different pHs, so that we can actually exercise the cells in the lab while witnessing the changes in pH because of the fluorescence levels of the nanosensors that are within the muscle cells. So when doing that, we're able to see, and these are CFS cells here towards me, and you can see in the very first panel that there are very high levels of acid, and when we add a drug called dichloroacetate into the laboratory system, the most important thing for us was that these abnormalities of acid are or appear to be reversible with a drug like DCA. Now, unfortunately, DCA is a bit like using a sledgehammer to crack a nut, and now we have a PhD student funded by Action for ME who is currently replicating these experiments and beginning to tease out where in the metabolic chain the abnormalities might lie, with a view to helping us develop drugs that may be applicable to human trials. Now, let me see whether I can get this one to work. So the other major... this might not work. Whoop. The other major organ that you'll remember from the vascular system is the liver. And the liver is a major capacitance vessel. That one's not going to work, I don't think. In the human. And in dogs and animal experiments, it's been found that the liver acts as an enormous sponge and contributes significantly to blood pressure control. So one of the things that we speculated was that the liver may play a role in maintaining that head of steam that we are so keen to explore and investigate in patients with CFS-ME. And what we've been able to do using MR technologies is ask patients to perform a Valsalva while we are looking at measuring their liver volume. And if the videos had worked, you would have seen that there are dramatic changes in the size of your liver in response to the different phases of a Valsalva. What James was saying earlier on about how hard the analysis of these experiments can be, so far we've done 55 of these scans in patients with CFS-ME, but so far it's taking medical students that we employ over the summer holiday a week to analyse one patient's scan at the moment. So we need to progress the automation of this very rapidly. But essentially what this shows is that this is a control patient, and physiologically we would expect liver volume to increase through the different phases of Valsalva. And you can see in the CFS-ME patients that there is a consistent decrease in liver volume in response to the various different phases of the Valsalva. So this is an area of particular interest for us at the moment and may explain why the head of steam is not as high in patients with CFS-ME. The third MR area that we're particularly interested in is cardiac MR. So the heart being right at the centre of that autonomic cardiovascular system. And we've done a number of series of patients now where we've been looking for structural abnormalities using MR and haven't found any structural abnormalities to date. But MR allows you to look at bioenergetic function of the heart, and we measure that using something called a PCR-ATP ratio. And ordinarily, a ratio of less than 1.6 would be consistent with a cardiomyopathy, a subclinical cardiomyopathy. You can see there's a considerable spread here in values in our 20 patients with CFS-ME. And the long and the short of it is that a third of the patient group that we've studied have a value less than 1.6. If we use some novel technology called tagging, where we apply over the scan a network of magnetic beads, and as this is the left ventricle here, and if you watch the beads as the left ventricle is contracting, what you're able to see is that there is some torsion about the left ventricle in patients with CFS-ME. So ordinarily we would expect the heart to pump in a very rhythmic, systematic way, but what appears to be happening in CFS-ME is that there is an exaggerated torsion of the left ventricle as the heart pumps, which translates into problems functionally downstream in terms of cardiac output and stroke volume, which we've been able to confirm in patients with CFS-ME. So in summary, what I hope I've illustrated to you is that we've confirmed that there are autonomic abnormalities in patients with CFS-ME, and the message to you as a CFS-ME audience is that those are consistent abnormalities in this patient group, but also in patients with fatigue-associated diseases. So we've done similar experiments in patients with primary biliary cirrhosis and autoimmune liver disease and found similar and consistent abnormalities. We've also found that there are brain, cardiac and muscle abnormalities in patients with CFS-ME and in patients with fatigue-associated diseases. And again, lessons that we can learn from in a bidirectional way are very important to advance our knowledge around CFS-ME, but also fatigue-associated chronic disease. So in conclusion, I hope you'll agree that fatigue is a very common symptom. It's the commonest reason why people go to see their GP. It affects a huge number of people with not only CFS-ME, but also chronic diseases. There are clearly emerging abnormalities, physiological abnormalities that I think we've been able to illustrate in our work from Newcastle. And symptoms and objective abnormalities of the autonomic nervous system appear to be a consistent finding and may offer a target for symptomatic treatment for patients who present with fatigue. It's very important for me to acknowledge the considerable support that we receive from a range of different funders. We're very lucky to have a range of charities who are very active supporting ME research in the UK. Action for ME have supported a number of PhD students for us. ME Research UK are extremely generous to us, and we've had some support from the ME Association and a number of grants from the Medical Research Council. So we're very grateful for all of their ongoing support. And final thing, if you're interested in our research, I've progressed into the 21st century, and we now have a research Facebook page, and we've got over nearly 1,500 followers on the Facebook page. So do come and pay us a visit, and if you want to know more about the research that we're doing. So thank you.
Thank you very much, Julie. Thank you. Okay, one or two questions perhaps before tea. Yes, two down here, yes.
Would the fatigue that you describe in different conditions also include post-exertional malaise?
They do, yes.
The same quality?
Yeah, absolutely. It's not of the same severity as in CFSME, but of the same quality. Okay. So it's very much the same descriptions of, I feel like I'm a torch where the battery's dying, you know, the brain fog, very similar characteristics, but in some of them, the severity is not as severe as CFSME. Sjogren's, the severity is equal to CFSME, and PBC in a proportion it's as severe, but the range is different.
Okay, thank you.
The gentleman behind. And we must move on, we're really pushing it. Yeah.
The increased acid, you said, in the brain, I don't see why that's related to the autonomic system, because surely it's a result of increased glycolysis and decreased oxidative phosphorylation.
Yeah, so it may well be. So the increased acid in the muscle that we found, there is a relationship between the autonomic nervous system severity and the abnormalities of muscle that we found. And we suggest that that is because some of the transporters that remove acid from within the cell are controlled by the autonomic nervous system, and the runoff via blood vessels, exercising muscles, is again controlled by the autonomic nervous system, which may explain why there's a relationship between the autonomic abnormalities. But you're absolutely right, the underpinning abnormality is much more likely to be related to abnormalities of biochemical function or mitochondrial function.